enhancement through drug permeability across the membrane. As we discussed earlier, that in order to improve the drug permeability, we need to have lipid solubility. So the drug which have more lipid solubility will can easily be crossing the membrane. So on several occasions, the rate limiting step in the drug absorption is transport through intestinal epithelium owing to the poor permeability. So if we try to improve the intestinal permeability, there is a chance that the permeability of the drug and in turn viability of the drug can be enhanced. So with an increase in number of emerging hydrophobic drugs, several lipid-based formulations have been designed to improve their bioavailability by a combination of various mechanisms briefly discussed in the following slides. So what different formulations, lipid-based formulations are there which will in turn improve the bioavailability? Lipid solutions and suspensions, micellized solubilizations, porous emulsions, microemulsions, multiple emulsions, self-emulsifying drug delivery systems, self-microemulsifying drug delivery system, self-nano-emulsifying drug delivery systems, nanoparticles, and liposomes. So what are the different mechanisms involved in order to improve the permeability? Physicochemical mechanisms and physiological mechanisms. In physicochemical mechanisms, we can enhance the drug dissolution and solubility. In physiological mechanisms, we can enhance lumen solubility by stimulation of secretion of bile salts, endogenous biliary lipids, including phospholipids, cholesterols, which together forms mixed micella and facilitates GI solubilization of the drug. The second technical mechanism of enhancing permeability is by reducing the gastric emptying time and thereby increasing the time available for the dissolution and absorption in the stomach. And the third mechanism is increasing in intestinal membrane permeability. Then we have another mechanism, decreasing the intestinal blood flow, then decreasing the luminal degradation of the drug, then increasing the uptake from the intestinal lumen into the lymphatic systems and reducing first pass hepatic and GI metabolism. These are the different mechanisms by which we can enhance the drug solubility. We can enhance the drug permeability. So, what are the advantages of this kind of formulations, lipid-based formulations? Physicochemical advantages are there. Pharmaceutical advantages are there. Pharmacokinetic advantages are available. Pharmacodynamic advantages are available. Physicochemical advantages such as solubilization of drug with low aqueous solubility, then solubilization of lipid drugs against hydrolysis or oxidation. Then in pharmaceutical advantages, better characterization of lipid excipients, formulation versatile and the choice of different drug delivery systems, opportunity for formulation as sustained release products. Then pharmacokinetic advantages, improved understanding of the manner in which lipid enhances oral bioavailability. Then reduced plasma profile variability, and potential for drug targeting applications. So even we can achieve drug targeting by lipid-based systems. Then pharmacodynamic advantages such as reduced toxicity, consistency in the drug response. Some lipophilic drugs such as steroids have applicable solubility in triglycerides alone. It is therefore comparatively straightforward to administer the drug in an oily liquid and thereby achieving satisfactory absorption. One advantage of this formulation approach, however, is that oil alone rarely provides the solubilizing power to dissolve the required dose in reasonable quantity of oil. The limitation of this option for using simple drug or by oil formulation is basically this limitation. So lipid solutions and suspensions. The ability, first we'll discuss, this is about lipid solutions and suspensions. Then solid lipid nanoparticles. To overcome the disadvantage associated with the lipid state of the oil droplets, the liquid lipid is replaced by the solid lipid, leading to the formation of solid lipid nanoparticles. In contrast to emulsion, the particles consist of a solid core made from solid lipids. They are characterized by mean diameter between approximately 100 to 1000 nanometers. There are two basic production techniques for solid lipid nanoparticles: homogenization of melted lipids at elevated temperatures and homogenization of suspensions of solid lipids at or below room temperature. Then we have another technique of liposomes. Liposomes are broadly defined as a lipid bilayer surrounding an aqueous space. Liposomal drugs can be embedded in the fatty regions, while hydrophobic substances are held in aqueous internal spaces of these globular vesicles. Then we have another technique of ion pairing. The ion pairing approach involves co-administration of hydrophilic other polar drugs with insoluble lipophilic counterions, which consequently improves the partitioning of the resultant ion pair relatively more lipophilic into the intestinal membrane. In fact, the approach seems to increase the oral bioavailability of ionizable drugs such as ethanol by approximately 2 folds. However, it is important that a counterion possesses high lipophilicity, sufficient aqueous solubility, physiological capability, and metabolic stability. Then compounds which facilitate the transport of drug across the membrane are called permeation enhancers or the permeation promoters. This method is used mainly in the case of hydrophilic drugs, which are expected to have difficulty in penetrating the lipid structures of the biomembrane. Penetration enhances act interaction and act interacting into the lipid parts with the polar components in membrane phospholipids. So these are the different techniques which can enhance the permeability of the drug.